So ready?
Ready to go.
Okay, good, thank you. All right. Good morning, afternoon, evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us, the 29th seminar, and I call it ACS Synthetic Biology Day because we have the founding chief editor today. I just came back Tuesday from Portugal, where I met many SynBYSS people and new SynBYSS speakers in an ECI conference. And escaping from the reality, I felt as if I am in heaven, meeting potential collaborators and spending a weekend in Lisbon. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor Chris Voigt. He does not need any introduction because we know, all of us know him. He's of course the professor at MIT, the chair for MIT-Broad Foundry, and the founding editor-in-chief of ACS Synthetic Biology for a decade until he just stepped down recently, and also my postdoc advisor. He's one of the geniuses I met and a true pioneer with the seminal paper of bacterial photography that made the synthetic biology community excited in 2005, including myself. He's also the founding chair of the SEED conference and came back as the chair of this year's SEED conference in May. So Chris, the podium is all yours. Thank you so much for joining out of your busy schedule.
Absolutely. Thank you, Tae-suk. So I think this is a fantastic platform and it's a great opportunity for many people. So I'm Chris Voigt, professor at MIT, run the Synthetic Biology Center here. And I just wanted to use this 5-minute time to describe how we want to engineer the Earth, not just model organisms, but everything that's living here. So if we look at what's known out there, so all of the amazing work from the human microbiome, understanding the guts that are on and within us, has led to these technologies that allow us to completely map the microbes associated with an environment. And it's remarkable what we don't know. So it's been estimated that there are 10 to the 12th different species on Earth, of which we've only described about 10 to the 6th. So we're 6 orders of magnitude away from describing all the bacteria that are out there. I mean, that is a remarkably small fraction of what's available. And it's an even tinier fraction if we look at what can actually be engineered. So 10, 100, we could argue about the numbers, but there's a limited number of model species that we're really good at engineering compared to what's the capability is. And so bacteria are everywhere. They're not just on our body and on our skin, but every little surface and every little subsurface on Earth is filled with microbes. The Air Force looked at many of the microbes associated with some of their airplanes, and there's specific communities, not just associated with the seats and the walls and the ceiling, but also in the hydraulic fluid, in the gas tank, and so on. Every wall, every key of your keyboard has a different community of bacteria on it. So we want to know what's out there, how we can get those microbes out, how we can engineer them to have a function, and then put them back in to carry that function in the field. And it's absolutely remarkable what the capacity is. You know, we look at the gunk that's growing in your bathroom wall as just gunk, but it's incredibly powerful gunk. So 1 teaspoon of bacteria has more DNA storage capability than the entire internet, more logic gates than 2 billion Xeon processors. So it's a computing and data storage powerhouse. But it's not just computing, it can also make things. It can build its weight, it can fix itself. It has 800 billion more genes than the human. It's self-healing, it's self-powering, and so on. We just don't know how to access all that. And so it comes across as gunk on the wall. So with SynBio, we can start to pull out those species and do all sorts of things with them. We can put in sensors, that respond to different chemicals or conditions. We can do computing within the cells if we can access it. And we can get all the different things from the making of materials, the breaking down of materials, the communication with other cells, communication with electronic systems, and so on. We can store whole movies in the microbiome. And so everything I'm currently focused on these days is about how to create technologies that allow us to do these capabilities from SynBio, but to do it in real organisms and not just model ones. And we've been showing the impact of this. So one example is some of the students from my lab took a bacteria out of soil, engineered it to deliver fertilizer to corn. And that's now on millions of acres of US corn and is reducing fertilizer usage and having a real impact on greenhouse gases. This was spread on as much area as the state of Massachusetts almost. We can also take out communities of bacteria that are part of living materials. And this is an example where we took many of the microbes associated with making architectural materials and engineered them to carry pathways that would change the color of the blocks. This is human melanin that was put into the pathway was put into a microbe that's a contaminant of the process to make these bricks. They're substitutes. It turns the bricks black, protects them against UV. And then we took some microbes off the back of a frog to engineer them to have an antifungal that was killing the frogs and put them back in. So these are all examples. These are all crazy microbes out in the environment that had to be pulled out of these complex communities, engineered to have a valuable function, replaced to those communities to carry out that function. And the potential of this is extraordinary. We're just touching the surface of it. So if you look at ships out at sea that are corroding in salt water, there are microbes that accelerate that corrosion, inhibit that corrosion. We want to pull those out, engineer them, put them back in. We want to be able to use microbes as part of food production in ways that are unimaginable now, and start to integrate them into consumer products. So maybe in the future, there are microbes that are stopping you from smelling bad or getting rid of your perspiration or causing insects to avoid you, or do all the things that we currently associate with chemicals and petroleum. So that's the 2 cents of what my lab is up to in 5 minutes these days, and very much look forward to meeting as many of you as possible in the future at one of these meetings that are now starting to come online and be in person again. So with that, thank you, Tae-suk, for giving me my 5 minutes, and I look forward to seeing your talk, Ruben.
Thank you so much. Wow, amazing talk as always. I mean, you are always a visionary, and I love it actually. Also, I believe I brought billions of microbes or 10,000 species from Portugal this week because of my travel over there. So thank you so much. I mean, and so now, the main speaker of today with a longer introduction, and Dr. Ruben Uribe is a project manager at the Novo Nordisk Foundation Center for Biosustainability in Denmark. And coming from Mexico originally, he has developed his scientific career working and studying in the US, Netherlands, Germany, UK, and Denmark. He obtained his PhD from the Technical University of Denmark in the group of Professor Morten Sommer. He helped to set up the foundation for CRISPR-Cas antimicrobials, resulting in multiple patents and spin-off company called SynBYSS. And also, his current work focuses on the intersection between the microbiome, Chris just talking about today, and synthetic biology, developing the next generation of microbial therapeutics. So I believe he gave the nice introduction for his talk today. His scientific work has resulted in various patents, as I told you earlier, and publication in the area of synthetic biology, gut microbiome, and CRISPR. He has a personal passion for gut microbiome again, and then fermented food. So actually visiting him in July for the seminar, I hope he giving some fermented food for my dinner or lunch, hopefully. And then at home, he produced his own yogurt. I love yogurt, and sourdough and bread and various other fermented food. So I'm looking forward to visiting you. And now, take it away. I mean, this is all yours. And thank you so much for your time from Denmark.
Thank you, and thank you for organizing this. I think it's really a great platform. Yeah. So I will start now, and let me see. Can you see my presentation?
Yes.
Perfect. Yeah. So yeah, I'm gonna talk about some of the work that I do to engineer the human microbiome. I think the introduction that Chris gave actually, like, it's pretty a good match to what I'm going to present today. My name is Ruben. I am co-principal investigator of the Bacterial Synthetic Biology section at DTU at the Novo Nordisk Foundation Center for Biosustainability in Denmark. Yeah, so for the ones that don't know, I mean, that is located here in Copenhagen. I mean, and it's actually, I mean, an island where Copenhagen is located. And just a little side note here, it's a really nice place to live. If you haven't been to Copenhagen, and I know that Thai is visiting us here in July, so I'm really looking forward, like, for his visit. Yeah, but this is a really nice place to live. And I mean, when the weather is nice, we are usually out enjoying the city. But during the work hours, we are usually here at the Novo Nordisk Center for Biosustainability. Our group works in different areas of synthetic biology. We work in developing microbiome therapeutics, drug resistance, studying drug resistance and collateral sensitivity, developing sustainable food production, and studying genetic heterogeneity. However, I am leading the group of microbiome therapeutics. And as Ty mentioned, like, I have a huge passion for the gut microbiome. And yeah, I mean, I like to show this picture because I think that, I mean, in the last 2 decades or so, we have started to realize how impactful it is, the gut microbiome, in our health. It is usually estimated that there are trillions of bacteria and fungi and phages, viruses, and archaea that live in our body, and they exist in these type of interactions, which some of them are actually very beneficial for our health. People have identified that the human microbiome can affect different aspects of health, like obesity, the way people respond to cancer treatment. There has been studies associating the human microbiome with depression and anxiety. So it is a really interesting therapeutic target. In that sense, in particular, the gastrointestinal tract contains most of the gut microbiome in our body, and it's there where most of my research focuses on. And what I find very interesting about the human microbiome is that it is essential for our health. It is something that we are not born with. Like, we acquire this as soon as we are born, but the main, and sometimes I like to think about it as almost like an organ in our body. However, the main difference with this organ is that it doesn't have any of our DNA. Instead, it's thousands of different species that form, of microbial species that form this organ. And as I said, like we as soon as we born, we are first exposed to these bacteria that start to train our immune system and help develop our brain functions. It is an extremely important part of our development, and it changes throughout life. It's never the same composition throughout different parts in our life. Our gut microbiome will continue to change. Some species will be lost. Some species will join the microbial community. However, we are only starting to understand what are some of the factors that affect the composition of the gut microbiome, and there are many. Antibiotic usage, the mode of birth, your diet, your genetics, if you have a pet at home, your environment, if you live in a rural community or in an urban community, where specifically in the world do you live, if you have siblings, with your partner, like all of these things will affect the composition of the gut microbiome throughout your life. And what we would like to understand and to learn to be better at is how we can harness the gut microbiome to treat different diseases. How can we produce microbial therapies? And for that, I mean, I think there are different approaches. I mean, people have tried to use fecal matter transplants, for example. However, I mean, I don't think that is I'm personally not a huge fan of that, because in the end, you don't know specifically what is that you are adding that is improving the health of the patient. It is in a way, I call it the brown box, because you are just adding a community of complex bacteria, but you don't really know what is giving the effect. My goal is to develop microbial therapies in a more targeted manner, to identify what are the mechanisms that a specific bacteria or a community of bacteria do to improve the health of the host. And using synthetic biology, enhance those abilities. Yeah, so the focus of my research group, it's in the interaction of synthetic biology and microbiome research. We try to develop microbial therapies to treat diseases in a transformative and targeted oriented manner. And I mean, to do that, I like to think that there are two approaches. You can either reduce or eliminate something from the gut microbiome, or you can add or enhance something that is already there. And I will present two stories from some of the work that we are doing in these areas. First, I'm going to present some of the work that we have been doing to reduce or eliminate some of the elements from the gut microbiome. And for that, I mean, we started with a question. So there is, how can we modulate the composition of the gut microbiome? We know it's a very complex community, and we know that there are good and bad players. But how can we develop a tool that allows us to specifically remove the bad players from the complex community while leaving the good ones intact? And to do that, we came up with the concept of CRISPR-Cas antimicrobials. So the idea, it would be that it is to deliver a DNA vector with the instructions to express a CRISPR-Cas system to do self-targeting in the chromosome of the bacteria. When there is this DNA break inside the bacteria, I mean, most of them they are not able to repair it through non-homologous enjoining. Instead, most of them, they use homologous recombination. However, if you overwhelm that repair pathway, it usually results in death. So in brief, the idea of the CRISPR-Cas antimicrobials is to use a CRISPR-Cas system to program the cells to commit suicide. And the beauty of it is that, I mean, we already have different types of antimicrobials. So for example, we have the antibiotics, which they are sort of completely unspecific to some degree. When you throw them into a complex community, they just kill everything that it can hit. Then you have phages, which are a little bit more specific, sometimes a strain to the level of a strain specific. However, the beauty of the CRISPR-Cas antimicrobials is that they allow for to the level of precision that is at the sequence level. So you can use the CRISPR-Cas system to target specific sequences, which could be used for different applications. They can be used to treat infectious diseases, to remove antibiotic resistance genes, to do in situ microbiome engineering, or for tailoring of the gut microbiome community structure. And so this is some of the first work that we did around this concept. So as I mentioned, the concept is to develop these vectors that express CRISPR-Cas9 and that have instructions in the cell to commit suicide. And in the beginning, when we did this experiment, so for example, here I did this experiment in an inducible system using 2 strains of E. coli, K12 E. coli strain, and the probiotic E. coli measles. And I showed that, for example, the CRISPR antimicrobial was specific to only kill the E. coli K12 strain. However, what I was interested about is what were these cells surviving here. I wanted to learn more, how these strains managed to survive and how I could manage to improve more the CRISPR antimicrobial to reduce these escaper cells. So we started to sort of break down the different ways in which the bacteria can escape the CRISPR antimicrobials. I mean, here in this side of the slides, I show our ideal scenario, what we wish it will happen. The phage delivers the DNA, the bacteria expresses the CRISPR-Cas9. CRISPR-Cas9 forms a complex with the Cas9 forms a complex with the guide RNA, and then they target the chromosome of the bacteria, which would result in cell death. However, there are some modes in which the bacteria might be able to escape. One of the most obvious is that it might develop resistance to the DNA delivery. Other ways in which it could develop resistance is by inactivation of the Cas proteins, or by inactivation of the expression of the Cas proteins or the guide RNA. It can also happen that the bacteria can acquire a mutation in the chromosome in the target site, so the CRISPR antimicrobial cannot work anymore. And there is also the issue that when you make the double-stranded break, the cell will try to repair that break. And this will be a continuous fight between you trying to kill the cell and the cell trying to survive. So we wanted to understand better how we could, what were the most common mechanisms of resistance and how we could overcome some of them. And to do that, I mean, as I mentioned, our hypothesis is that if you can overwhelm the homologous recombination repair system, then you should be able to improve the cell death. And one of our hypotheses there, it was to see if the level of Cas9 would affect the killing potency of the CRISPR-Cas antimicrobials. So we constructed 3 different vectors with different promoters to modulate the level of Cas9. So for that, we had 2 targets. So we had a control target that is completely random, is not targeting anything in the chromosome of E. coli. And then we have 1 target that is in the chromosome of E. coli. And then we have 3 different promoters, a weak, a medium, and a strong promoter. And what we could see is that, there was sort of a correlation between the level of expression of Cas9 and the potency of the CRISPR-Cas antimicrobials. Next, we wanted to find out if there would be a better effect by having multiple targets in the chromosome. So what we did was that we searched for sites in the chromosome of E. coli that would be repeated multiple times. This way, by using 1 guide RNA, we would target multiple sites at the same time. And the idea would be that if you target 1 site, it would be less toxic than targeting 2 or 3 or multiple sites in the chromosome. And we actually found some sites that are more than 20 times in the chromosome of E. coli. So we expected that those guide RNAs would be extremely toxic and would kill most of the E. coli's. However, what we found was that that was not the case. There was actually no correlation between the number of target sites. There was also no specific effect between targeting a non-coding region versus a coding region. And there was also no improvement by targeting essential versus non-essential genes. So at this point, what we concluded from this experiment was that the level of Cas9 is important, but the number of target sites is not as important. There seems to be other ways in which the level of potency of the target site is regulated, which is still not completely clear what it is. But yeah, I mean, we moved forward with this project, and we wanted to find out now, okay, so we have more Cas9, and this makes it more potent. We have some pretty potent guide RNA, so these targets here, and we know that that we still have some bacteria surviving. So we wanted to find out what these bacteria are doing. So we designed an experiment to see if this killing was in Cas9 or it was somewhere else, if it was a mutation acquired somewhere else in the chromosome that would allow for the cells to escape. So as I show in this diagram, first we had our Cas9 being expressed and cutting in multiple sites. And from this, we would have 2 outcomes. In one outcome, the cell would die, and in the other outcome, it would escape. And all of a sudden, the CRISPR-Cas antimicrobial would not work anymore. So what we would do is that we would reintroduce a plasmid with an intact Cas9 and a different resistance marker to select only for that plasmid. And what we could see is that most of the cells became sensitive again. Sorry, only one of the targets became sensitive again, and this was a target that has a single cutting site. So what we saw was that the other targets, so the ones expressing multiple targets in the chromosome, would be sensitive again to the CRISPR-Cas antimicrobials, while for this specific target, which is found only 1 time in the chromosome, the bacteria, some of the colonies that we collected were still able to escape this killing. So we sequenced some of these bacteria to find out where were some of these mutations. And we found out that most of the mutations were actually accumulated in Cas9. And what we learned from these experiments is that in general, like when you have optimized the level of expression of Cas9 and you have multiple target sites, the preferred target for mutations will become your Cas9, but this can be overcome by introducing a second Cas9. So to having some redundancy in your genetic circuit. So what we can summarize from this project was that in order to maximize the effect of CRISPR-Cas antimicrobial, it is required to have high expression of Cas9. The potency of CRISPR-Cas antimicrobial does not depend on the number of target sites or function of gene targets. However, having multiple chromosomal targets, redundancy, was a successful strategy to reduce mutations at the target site. E. coli can acquire mutations that allow it to escape the antimicrobial effect. However, SpCas9 being the preferred target for the mutations. And these resistance mechanisms can be overcome by reintroduction of an intact copy of Cas9. So until this point, what I described was some of the projects where we have worked in technologies to modulate the microbiome by eliminating or reducing the abundance of a specific species. However, there is also another approach. There is the approach of adding a species or enhancing the therapeutic potential of a gut bacteria. And for that, I mean, we have the concept of microbiome therapeutic, which is engineering different circuits, genetic circuits to sense and process the information from the environment. And the idea is that we can couple different biosensors to sense the temperature, pH, oxygen, or internal metabolites of the bacteria, process this information with using different genetic circuits, and then the cell should be able to take decisions of what to do in a specific condition. So, for example, produce a therapeutic or activate a pathway for degrading a specific compound or enzymes, or to target a different species and modulate the specific community, or a kill switch, so that, for example, if the bacteria detects it's not in the right environment, then it can commit suicide, sort of like to contain itself to a specific environment. And to do that, it's actually pretty challenging because not that many people have tried to do that. So we have identified some key challenges to develop these microbiome therapeutics. So one of them is to choose the right chassis strain. As Chris mentioned in the beginning of the presentation, I mean, I think we have been focusing in mainly in model organisms like E. coli or Saccharomyces cerevisiae, but there are thousands, or actually millions of other species that have the potential to have a therapeutic effect in the human body. So one of the key challenges is to be able to isolate and identify some of these bacteria. However, that is only the beginning of the story, because once you isolate them and identify them, you also need to develop genetic engineering tools to work on them. And I mean, if you have only worked with E. coli, you might not be aware that most of the tools, plasmid vectors, promoters, all of these things that we use for E. coli do not translate completely into other organisms. And more importantly, they have only been studied, well, they have mainly been studied in the context of a fermenter in a lab. They have rarely been studied in an environment that is more, that is closer to the gut environment. Another challenge is that we don't know of that many elements to modulate the gene expression of some of these organisms, or for a specific environment, like in anaerobic conditions or in the environment of the gastrointestinal tract. We also need to develop different strategies to improve the safety of these microbial therapies, as well as the biocontainment. because it is a concern. If you're going to put a live therapeutic into your body, you need to make sure that it only stays in the patient that is supposed to treat, and that is not there in the environment producing that therapeutic function somewhere else. You also need to develop different approaches to increase the viability and the colonization of the gastrointestinal tract. And it is a particularly challenging point because you're like understanding what are the factors that affect colonization. It's very complex. There are factors that connect with diet. There are factors that connect with the immune system and with the genetics of the host, and as well as like the composition of the microbiome community. However, there are approaches in which you can engineer circuits in your chassis, so that is able to use a specific carbon source or colonize a specific part of the gastrointestinal tract. And we have also projects working on that. Another important part is the genetic stability, because the main difference between inert drugs, is that these are live therapeutics. And if you program something, if you program a function in your live therapeutic, like for example, producing a specific therapeutic, it has to produce that one in the host. It cannot mutate, it cannot change the therapeutic that is producing, because that is simply not then a very reliable way of treating diseases. So studying approaches to increase the genetic stability of the strains and improve this is also a challenge that we have identified and that we're working on. We are also developing different in vitro assays to evaluate our microbial therapies. An important part of developing a therapy is that you have to do animal studies to test the function. However, this cycle can be pretty difficult, because doing animal study takes usually several weeks, and processing the data, and all of these things, plus the ethical considerations of using animals for these studies. All of these things have to be taken into consideration, and that's why we also put a lot of focus in developing in vitro assays that allow us to have some readout of our different strains, how much we have optimized them, how much we have changed them, to actually have a positive effect in our model. So from these in vitro assays, our hope it is to identify the best candidates that we can move into animal models, so that we can study their effect in an animal. And that is another part that in our lab where we work, developing animal models and identifying biomarkers for the different therapeutics that we are developing. And of course, before you go into humans, an important part is that you are able to scale up your production, because one thing is to produce it in a test tube or to produce enough for an animal study. And a completely different thing is to scale up the production and manufacture of your microbial therapy for applications in humans. So we also have one project that is focused in that part of developing the microbial therapies. For the rest of the presentation, I will focus in a specific point on the work that we have done to develop elements to modulate gene expression in situ, in particular promoters. And for that, I think that if you have worked with E. coli, you know that you can use certain constitutive promoters. However, most of the constitutive promoters that were available in the literature, they were mainly characterized in lab conditions. So what we wanted to do is to engineer genetic circuits in microbial therapies, but using more robust modular regulatory elements. So for that, we created a library of sigma 70 promoters, and we characterized that in aerobic and anaerobic conditions, and in a mouse, and we measure the level of expression in these different of these promoters in these different conditions, and identify from these specific promoters that have robust, reproducible level of expression between the different environments. So the idea would be that if you generate a genetic circuit in the lab and you characterize it there, when you put it in an animal model, you should be able to do it in a more reliable manner. So we found these promoters that behave basically the same in aerobic and anaerobic conditions and inside the gastrointestinal tract of a mouse. So then we wanted to use these promoters for a specific application. So we chose to use the promoters to produce different therapeutic peptides. And I will now give a little bit of an introduction to why we chose peptides and what a specific peptide which targets the GLP-1 receptor. So I'm going to give an introduction to obesity, which is a pretty common disease in Europe and in North America. I mean, in the European Union, overweight affects 30 to 70%, and obesity affects 10 to 30% of adults. Obesity is a leading risk factor for type 2 diabetes. About 60 million people in the European region have diabetes, 10.3% of men and 9.6% of women. Health expenditure for diabetes was estimated at $105 billion in the European region in 2010, and by 2030, this expenditure is expected to reach $124.6 billion. Worldwide, obesity and overweight-related diseases kill about 3.4 million people annually. Almost 80% of these deaths occur in low and middle-income countries. The WHO projects diabetes death will double between 2005 and 2030. So this is a very complex disease. And I think that most of the people, when they hear about obesity and overweight, they think that for a lot of these people, simply changing diet or exercising more is enough. However, like the picture in the medical community is changing. Like now people start to see obesity and overweight also as that genetic play a very important part in this disease. And for that, there are also very specific and effective treatments to treat this type of diseases. So one success story is the GLP-1 receptor agonist, which is these small peptides. GLP-1 is a hormone that is naturally produced in the body after meals. And when it's produced in your body, it helps to give a feeling of that you don't have that you are not hungry anymore. However, this peptide has a very short half-life of approximately 5 minutes. And this is because the body regulates these signals very tightly. And throughout the years, people have discovered other versions of these peptides with extended half-life. So for example, exenatide, which is a peptide found in a specific reptiles, has extended half-life of 174 minutes, and a chemical modification of this GLP-1 peptide results in a better product that is called liraglutide with 13 hours of half-life. And the advantage is that I mean these drugs are peptide-based. They promote weight loss and are associated with a lower risk of hypoglycemia. They have a really high demand. For example, liraglutide became the 163 most commonly prescribed medication in the US in 2017, and this is growing like crazy. with more than 3 million prescriptions. And I mean, the inconvenient part about this is that most GLP-1 receptor agonists are only available as injections under the skin. So for example, if you are using something like exenatide, you have to inject it 2 times a day. And if you're using something like liraglutide, well, there are different versions of this, but one of them is that you have to inject it once a day. There are other versions that you cannot, that you only have to do it once a week. No, nonetheless, like this is something that people have to live with for sometimes the rest of their lives. And I mean, having daily or weekly injections can really become a hassle for people that have to live with this type of therapeutics. So our motivation was to develop a microbial therapy to deliver some of these peptides. Why the oral route? I mean, it's because this is the preferred route for drug delivery. But I mean, the gastrointestinal tract is actually made to process all of these peptides and proteins. So it's not very friendly for this type of therapeutics. And there are many obstacles for this. So you usually, like the barrier, if you're a healthy person, this barrier is not very permeable. It will not allow most things to pass through. In addition, there is also different enzymes that can degrade your peptides, plus the enzymes produced by different microbes that can also degrade your peptides. So in general, it's not that easy to deliver peptides orally, and that's why, like previously, like these drugs like exenatide and liraglutide were mainly injected. However, an advantage of the gut microbiome is that it can grow very close to the enterocytes and produce some of these peptides that directly there, and you can use different strategies to even enhance the permeability of the gastrointestinal tract to deliver some of these peptides. So for that, we constructed a strain, an E. coli nasal strain. This is a probiotic strain that has been used for over 100 years. We made it streptomycin resistant so that we can use this antibiotic in the mice. So that we can create a niche so that it can colonize. We also tag it with GFP so that we can easily find it in when we're counting the colonies. And for the expression, we use the endogenous plasmid of E. coli nasal called PMUD1, where we clone our peptides together with a secretion tag. So that after they are expressed, they are secreted into the environment. And we use some of the constitutive promoters that I presented earlier, which allow us to characterize our strain in the lab and expect that it will have the same behavior in situ in the gastrointestinal tract of the mice. So we constructed this agonist, and we tested them in diet-induced obese mice, and gave 3 doses of these bacteria daily, and we followed the body weight, and we did a glucose tolerance test at the end of the study to determine if the glucose metabolism was improved in these obese mice. And the antibiotic treatment that we give with streptomycin allows our streptomycin-resistant strain to robustly colonize in the mice. So we performed this study, and I mean, first I'm just gonna talk a little bit about the colonization. So as since we introduced this resistance into the chromosome of E. coli nasal, we were able to have very high levels of colonization after a single dose. And pretty much this colonization stayed the same throughout the study. As you can see here, for both strains, one producing GLP-1 and another one producing XND4. We collected at the end of the study blood samples and gut content samples from the cecum of in the gastrointestinal tract of the mice, and tried to quantify some of the peptides. So GLP-1 is a little bit tricky to quantify because it has a very short half-life. However, here I'm showing the results for XND4. And this one, we could find a trend in the mice where we could see higher levels. However, I mean, it was not significant between the control strains and the XND4, both in cecum and in plasma. In addition, we were able to see a trend in the reduction of epididymal fat in the mice for the GLP-1 groups and the XND4 groups compared to the control and the negative control. However, like, it was also not statistically significant. And we also saw an improvement in glucose tolerance. However, in this case, we saw an improvement. It was also not significant. But what is interesting is that we saw an improvement also in the E. coli control strain. And what is interesting about this is that there was recently a paper where they show that E. coli Nisl by itself has an effect in glucose tolerance. So this sort of explains this positive effect that we already see just by giving the probiotic that has not been genetically engineered. Yeah, but I mean, these results, I mean, it's a still ongoing work. We are redesigning the animal model and improving the strain to produce more of the peptides, as well as trying different strategies to enhance the permeability and stability of some of our peptides. But this is just some of the preliminary results that we have until now. And although most of the results are not significant, at least they are starting to show a trend. And I hope that they also emphasize that developing some of these microbial therapies is more challenging than that we originally thought, because it's this concept in which we are trying to move something from the lab into real life application. And we start to discover all of these challenges that we try to overcome using synthetic biology approaches. And I would like to thank my postdoc supervisor, Morten Sommer, as well as the bacterial synthetic biology section for all of their work in these projects. Today, I only had the opportunity to present 2 of the projects that we have been working on, but if you stay tuned, you will see some of the amazing work that a lot of these people are now doing. And I would also specifically like to thank the microbiome therapeutics group from the section, as well as the Novo Nordisk Foundation Center for Biosustainability, and Gubra, which help us with some of the animal studies. Yeah, and that's it from my part.
Okay, so fantastic. So actually, I realized you and I have many common research interests and vision, common vision and common interest. So I really enjoy your talk. Thank you so much. So actually, I have so many questions, but I wait until others' questions are answered. So maybe I already saw one question in the Q&A. So I'll, so this is a question from Vishal. As someone who is new to synthetic biology, what do you suggest to get acquainted with the field? This is a more general question or career question, I believe.
Yeah. Yeah, I mean, I think one of the important parts is to learn what are the basic elements to regulate the expression of genes. So for example, get familiar to what is a promoter, what is a constitutive promoter, what is a ribosome binding site, I mean, and how you can use different tools and that are already available out there to optimize the expression of genes. I think that is a key part of synthetic biology. Yeah.
Okay. So any question from audience? You can raise your hand, or I see one hand. So now you can talk, Pistar.
Hi, thank you for the presentation. So one of the things that jumped out to me was like, since you're just starting with your experiments and most of the results are still not significant, how do you go about that, and how, like, what's your future goal on getting something that's more productive and that's more significant? And sometimes it may not be significant, but you know, everybody cares about something that's more significant. What's your future plan, and how do you want to proceed since you have the results that you clearly found was not significant at this moment?
Yeah, I mean, I think when we started with this project, we were a little bit naive. I mean, because we were only familiar with producing the proteins in the lab in a test tube. And I think that starting to get these negative results in the animal studies and all of these things, it has highlighted some of the challenges of developing this type of technologies. And from that, what we take, I mean, from these failed experiments, what we take is inspiration of how we can improve our technology. Of how we can improve our technology. I mean, I can only say that from those negative experiments, that has helped inspire new projects to try new things. So, I mean, it is frustrating sometimes, like to build a strain, prepare the animal study, go there, and then, you know, you look at your plots, you do your statistical analysis, and then the results are not significant. But, you know, you learn from these things. And I mean, that's what happens when a technology is new, right? Like you don't, we are building the foundation of for how other people might be able to use this experience to build better versions of what we are doing.
So wonderful.
I have two more questions. The first thing is about regulation in EU regarding genetic engineering. Like, I'm not sure how your project is affected by that, and what are the regulations that exist. And the second question is regarding the ethics and responsibilities as a scientist. So I'm one of the aspiring PhD student. I'll be studying synthetic biology, and what are some of the things that we can keep in mind when it comes to thinking about synthetic biology and helping human being, you know, in a way change their own capabilities when it comes to expressing different genes?
Yeah. I mean, so about the regulation, I mean, there is not a clear pathway for that, because as a new technology, I mean, you know, the authorities are learning together with the companies and like the different research groups, like what is necessary for testing this type of therapeutics. What I can say about it is that the picture is changing, and it's changing really, really quick. I think that 5 years ago, 6 years ago, it was, you know, the picture was completely different. Like there were very few companies working in this type of technology. And now it's, it's completely different. Now, for example, there is this company in the US, SynBYSS, for example, who, I mean, they already have some positive results from clinical trials. And this is probably one of the best examples of a synthetic biology company, because they are pretty far away in developing this type of therapies. And for me, it's very exciting to see that they are doing well, because they are sort of paving the way for other people, you know, that are still doing these animal studies, which are still not significant, but hopefully, we can change that soon. And about the ethics, I think that is also a very important question. I mean, because now we are developing genetically modified organisms that are doing functions that they are not there in nature. So we need to take this very, very serious, and being able to develop approaches for biocontainment. So that is probably something that is sometimes overlooked, like the biocontainment strategies. But I know that there are people doing research on that. I mean, Ty just published a really nice genetic circuit for biocontainment. And that is just one of the approaches, but like, it's another area of synthetic biology that we need to put a lot of focus on how we are going to make sure that that these therapies end up only in the patient, and then after that, they are contained only there.
Thank you.
Yeah.
Any other question from other audience? Actually, I have lots of questions, but I probably cannot ask the entire question right now. Actually, you made the great point. I mean, about the negative results sometimes I believe that sometimes lead to innovation, and unexpected results sometimes lead to innovation. So that's wonderful comment you mentioned. And I'll double check one more time. So no Q&A, no chat. In that case, I wanted to ask some questions, some technical questions. So first question is a very simple question. Have you compared the in vivo promoter data and the in vitro promoter data? You show the mouse promoter characterization, and then, but did you kind of compare that data with the just in vitro or the tube data?
Yeah.
And then that's correlated.
So what we found is, because we tried like over 8,000 different promoter sequences, that for the majority of them there is no correlation. So sometimes the level of expression that you would see in vitro is not the same that you see in the animals. But then from these sequences, we picked the ones that had the highest correlation between aerobic, anaerobic, and in vivo conditions.
So I believe the Gautam Dantas also did similar, work. I mean, published some work. I mean, about those kind of characterization, I believe. So that is kind of also kind of consistent, kind of with your data in the case, right?
Yeah. I mean, I think one of the main messages from that work was that you might spend a lot of time characterizing something in the lab. In aerobic conditions, but then when you put it inside an animal, it might just behave completely different, and that's one of the challenges of this type of technology.
Right. Sometimes, but there are some matching, kind of promoter between these two conditions, right?
Yeah. And that's also the point. But I think, I mean, for people working in this field, I think that it's important that you try to include some degree of characterization in multiple conditions. Like, it's not enough to do like just aerobic conditions. Sure. You should at least try to do something in anaerobic conditions, because that's where most of the bacteria will grow at some point.
Exactly.
Or at least that
Yeah, that's wonderful. So my second question is another kind of technical question or comment. So you use the streptomycin resistant research for the experiment, I believe, for your second part of the talk. And that would be good, but it would lead to antibiotic resistance spread in the real life kind of application.
Yes.
So could you comment on this?
No, we wouldn't use it outside of the lab. I mean, we only use it for the animal model because it allows us to create a stable colonization. Because otherwise, we would have to dose daily, and we have seen that this probiotic E. coli niso, it colonizes for a few days, but it washes out after like 2 or 3 days when you don't treat with any antibiotics. Right. So for us to have like a really high colonization and high numbers and reproducible between different mice, that's why we include the streptomycin, just to create a niche in the microbial community for our E. coli niso to colonize.
Yeah, but you also need to think about real, applications or real application, in human, or we do not have that high, colonization anyway.
But what I would argue there is that the transit time is also very different. I mean, humans are not mice. So, for example, a mice might poop the dose that you're giving between 6 and 8 hours. It will start pooping most of it. Right. While a human, depending on the gender, could have like more than 48 hours in which it will pass through. So the dynamics of colonization are also different between these two. And and I think when you have to choose, these animal models, you have to choose what you want to focus on. And in our case, we want to see like, okay, best case scenario, our bacteria can colonize really high numbers consistently. Okay, so we can do that. Now, is it expressing the peptide? Is it, can we detect it? Is it having the right function? Yeah. So it's just to simplify the model, because otherwise it becomes too complex.
Yeah, exactly.
Yeah, but we're aware we don't want to put antibiotic resistant genes in people.
Yeah, of course. Great. So let me double check one more time. So we don't have any Q&A now. There's no new questions. Okay, so let me close. So thank all for joining and staying today. We'll meet again next week on April 21st, Thursday, the same time, the same Zoom link. We'll have the Professor William Bentley at University of Maryland and fellow AAAS, ACS, and AIMBE, American Academy of Microbiology. Actually, I met him last week in Portugal, and we will have Young Huang at National University of Singapore. And as usual, the follow-up informal chat will occur without recording. So please stay here if you are interested in chatting with today's speaker and me, and I will promote you to panelists who can speak and show your handsome and pretty face. So thanks all of you, and I will stop recording. Thank you. Okay, stop recording.